you to all of the organizers for putting together this workshop and having the courage to actually hold it in presence. I was also not convinced that it's going to take place in presence, so thanks a lot. And yeah, for me, it's also been 2 years that I've been to Paris the last time. And so, yeah, it's a great place for mathematics and academic exchange. So I'm also very pleased to be here and present some work I've been doing in collaboration with Noemi David. And so it's on an incompressible limit for a Tumour growth model. And so the question that I want to address is a very fundamental one. So, so on the top row, you see two experiments, biological experiments. And so essentially here we have human embryonic kidney cells. There we have some tumor lines. So from my collaborators, Hideki Murakawa and Daniel Cohen. And so what we see is that as time goes by, we have these two cell populations migrating towards each other. So there's also a video. And so the question I want to address today is that as applied mathematicians, we always have a question to face, a choice to make, namely how we want to model, and in this case, how we want to model living tissue. And so on the left-hand side, I've just plotted some graphs, so lattice-based models where we essentially have individuals, so each cell is being described by its location on some discrete lattice, and then we prescribe some evolution on this lattice. Or alternatively, we identify each cell by its coordinate, and then we describe how the cells interact and then move according to surrounding cells. And so another choice is a macroscopic description, as we can see here, which instead of describing the evolution of each individual cell, describes the distribution of the cells instead. So how likely are we to find the cells at a certain location? And this is described by a continuous variable, like here. And then there's yet another approach, and that is the approach that I want to be, or I'll be talking about today, is this geometric approach where instead of tracking each individual, or instead of tracking the density of my cells, I describe my tumor, say, as a set that it occupies, and then how the set evolves as time goes by. And so historically, there's been a lot of work scaling limits of how you go from an individual-based kind of microscopic approach to a large scale, to the macroscopic scale. So for instance, by mean field limits or other scaling limits. And recently, there's been a lot of interest in how we can actually bridge this gap between a density-based description and this free boundary flow. And this is what I would like to address today. And so the very foundation for the model is, so we start with this macroscopic approach where the tissue is described by its density, which will be denoted by n of x and t throughout the entire talk. And I'd like to incorporate three mechanisms, essentially. So there's a pressure that avoids overcrowding. There's some drift, and this is the novelty of this work, and some cell growth and death. And so the equation that this gives rise to in the simplest setting is this continuity. Oh, I shouldn't be pointing. The continuity equation, essentially where we have this pressure term, so it's coupled through Darcy's law. We have some drifts, and we have these growth dynamics. And of course, there's different ways how we can couple the velocity to the pressure, and here we'll be interested in Darcy's law. So it's just essentially how you can imagine this is that cells will tend to flow downhill the pressure gradient in order to avoid overcrowding. And so in this talk, we'll be assuming this compressible pressure law. So essentially what you can see here is that as the density increases, the pressure grows, right? So the more cells you have, the higher the pressure on that tissue. And so we just choose this power law here. And so the incompressible limit consists of linking this macroscopic description, so this partial differential equation, with the evolution of this patch that the tumor inhabits, if you will. And so to fix ideas, I'd like to invite you to join me in a very nice computation, which I find quite fascinating. So we start with the power law, and then we just do a small modification. So you see if we cancel the k minus 1 here, then we have a factor of n to the k minus 2. So, I mean, these two lines are exactly the same. And then we continue. So we just multiply by a 1 in here. And so what you can see here, this term is exactly our pressure, right? By the way we chose our compressible or constitutive law. So let us replace this term by the pressure, as we've done here. And so in the final step, we just split this exponent on this guy, on this term, and on that term, respectively. Okay? So we've kept the k, so this fraction here, the n has moved there. And then we have, these fractions sitting here. And then p, the pressure, so this guy, raised to the k minus 2 over k minus 1. And so now the incredible thing that happens is as k gets very large, so as k goes to infinity, we see that this term is essentially 1. This term is also 1 raised to the power 1. And then we have p raised to the power 1. And so what that means is that if we have limit quantities n and p, they will satisfy this kind of complementarity relation that p infinity is equal to n infinity times p infinity. Or, well, if we rearrange the equation, we have this equation. And so this tells us something about our tissue. Namely, if the density is not saturated, so if the density is less than 1, then we do not feel any pressure, or the pressure is equal to 0. Or if we like to think about it the other way, if we have a positive pressure, then it means that our tissues or our cells are saturated. And so if we somehow have now like a link between this macroscopic description and macroscopic description and this free boundary, this patch of cells, namely the set where the pressure is positive, more or less. And so what's going on here is, so I've just taken this power law and I've plotted it for several values of k. And now you can see why this limit is called stiff limit or incompressible limit, because essentially the cells do not react to the pressure as k gets large unless our tissue is saturated. And so this is the limit that I'm interested in, because then we can make this identification. Okay. And so before we go into the mathematical details, I'd like to give a bit of a historical flashback, because essentially the topic started about 60 years ago with the well-posedness theory of the filtration equation, so by the famous Olejnik and collaborators. And then from there, Benilan and Crandall showed the continuous dependence of this nonlinearity, so this phi. So solutions will depend in a continuous manner on the nonlinearity phi. And so if you choose a power law, so if the phi behaves like a power law, then, in the 1980s, there's this British group that observed, by using formal asymptotic expansions, that as k gets very, very large, your density distribution actually looks like something that is flat. Flat like these mesas, so these mountain structures where you have a flat top, and then there's some small things going on, and then there's, again, a patch of where the density essentially is saturated. So these mountain structures are called mesas, and so this limit historically is called the mesa limit. And then, 1 year later, by Caffarelli and Friedman, they gave a rigorous foundation for this limit and justification. And so now the question is, so we've already got a bit of an intuition, but what's going on here? So why do we get essentially indicator functions, so sets or where the density is saturated, and then some other zones? And so the answer is, well, we have to study the equation. We have to have a closer look at just the pressure term in our equation, right? So I've just substituted the pressure in here. And so if we apply the chain rule, then we see that we can write this as a diffusion equation that has this nonlinear mobility. So k times n to the k minus 1. And so what does that mean? So whenever n is between 0 and 1, so whenever we're not saturated, the mobility is 0. And whenever n is larger than or equal to 1, the mobility is close to infinity. And so what happens is, in this limit, so we start with this gray curve. As k gets large, in an infinitely short period of time, we have that these mountain hills, quote unquote, will collapse. So mass will be diffused or transported, depending on how you like to look at it, to, well, the next possible space. And so what happens is that we have exactly these indicator type functions and then some density between 0 and 1. And so now that the thing is we want to identify these saturated zones with where the pressure is positive. And this is then how we build, this is our formalism of how we build the bridge between macroscopic and geometric model. And so one absolutely crucial instrument in this, in the mathematical justification is that we observe that this, the dispersal of the density driven by the population pressure, so this is this term, actually corresponds to a porous medium equation. So it can be written like this. And so there has been a very influential paper by Aronson and Benilan that gives you a very strong regularity result on the Laplacian, so second derivatives of the pressure, in the following way. And so you see that this bound depends on k. And so as k goes to infinity, this will go to 0. And actually, this result is very, very important, not just in the topic of incompressible limits, but also a lot of like fine estimates on the porous medium flow. And so this is one crucial tool that we'll be devising, and in our work that we've kind of extended or, well, find a novel approach to make it fit for our setting. So then the story kind of continues. So so after these fundamental works or seminal works, there was a paper by Gilles and Quiros where essentially they take a half space and then they have an injection at the origin. And so then they see how the free boundary propagates. And then, well, a couple of years later, there's the cornerstone or the stepping stone for our work, which is a paper by Perthame, Quiros, and Azquez, where they prove this Healy-Short type asymptotics for, in the context of tissue growth or tumor growth. So instead of having an injection that drives the free boundary, like in a Healy-Short cell, it's the growth dynamics of the tumor and the incompressibility that drives the growth of the tissue in this setting. And then from there have been various generalizations for various pressure laws. So there's this work by Hecht and Vauchelet for the singular pressure law that already incorporates some level of incompressibility. Then nutrients have been included in the dynamics by David and Perthame. And then also the strategy was extended to systems of interacting species. So for instance, normal and abnormal cells. In the case of the singular pressure law, we've also worked on this for the power law case. And then this was extended last year also to the full space. So up to this point, we only had one-dimensional results. And then quite recently, there has been another approach for non-monotonous growth rates, let's say, by Gillen, Kim, and Millet. And so what I did with Noemi was we wanted, so up to this point, there have been various generalizations, but we didn't have any satisfying answer to what happens when we include drifts to the dynamics. And so drifts are very important in tissue growth, you might argue, because, I mean, if we have, for instance, an oxygen distribution or nutrient distribution, this might induce a favorable, like a direction towards or a motion towards a favorable zone. And so this motivated the question of how we can treat this geometric limit in the case of convective effects. But still, I'd also like to say that this is a very, very active area. So there have been many people studying certain aspects of this model, and essentially there's three communities. There's the people from optimal transportation that have kind of a gamma limit approach to this type of problem. There's people working in viscosity solutions that give you a very fine description of the free boundary. And then, let's say the classical quote-unquote Sobolev theory that lets us describe, or that lets us derive the equation that is satisfied inside of our tumor patch. Okay, so as I was mentioning, the stepping stone was this model by Perthame, Quiros, and Azquez, where essentially we have these two effects, a pressure and a growth term. And so what they observed is that if we multiply the equation by p prime, we see that here we get an equation for p, and the equation looks like this. And so if we substitute our value for p, we see that we have a very large term appearing. So k is the quantity that goes to infinity that helps us establish the link between macro and geometric scales. And so as k goes to infinity, so this incompressible limit, we have exactly this, this Healy-Shor type problem. So whenever our pressure is positive, so inside of our set, inside of the tumor, we have to satisfy this elliptic equation. And this then will drive the boundary of our tissue. And then, so in their paper, they can also identify exactly the zone of saturation with the zone where the pressure is positive. And so what we did, as I foreshadowed, is we wanted to incorporate convective effects. And so now our model accounts for this pressure-driven dispersal. So if the tissue is too packed, it wants to expand. We have advective effects due to favorableness, so oxygen, nutrient gradients, etc. And we have the mitosis and apoptosis effects that also depend on the pressure. And so formally, again, we can multiply the equation by p prime. So actually, I've done a bit greater detail here. So we have p prime times dn by dt, which is a chain rule, so it's dp by dt. Here we have p prime times gradient n, which is a gradient p. We have another gradient p here, and then we have this p prime times n term, which if you write it down, it gives us this k minus 1 pk, right? Because this is a power of n. And so formally, if k goes to infinity, then only the last term matters. It becomes dominant. All the other terms vanish. And so we would expect to get this complementarity relation. So this is the equation that governs the pressure inside of our tissue. The question is, you want to pass to the limit here in a rigorous setting, so how do you get the compactness of, and what's needed here, the pressure gradient? Right? So you need very strong compactness results. And so this was the challenge, and this is what I want to tell you about today. So this is just one slide to show you, again, the connection to the Healy-Shor dynamics. So here we have in the zone where the pressure is positive, we satisfy this elliptic equation here. And then at the boundary, the pressure drops to 0. It's 0. And this then drives the evolution of the free boundary. So this omega is really an omega of t that grows. OK, so then, as I was saying, how can we justify this limit, k going to infinity, given that we have to have the strong compactness of the pressure gradient in order to pass to the limit in this term? And so there were some previous results in the literature on equations with drift, but they were not able to get this type of strong compactness. And the reason is that, in a sense, the Aronson-Benillan estimate, this classical parabolic result, fails, and it fails in a way that essentially the Laplacian of the pressure can only be bounded by a constant minus something that vanishes at 0. So it's essentially the problem that arises in this context. And so we wanted to circumnavigate this problem, and well, we came up with a different approach that uses essentially less assumptions than the few results that there are on the equation of drift. And we can still pass to the incompressible limit. So we can still rigorously justify how we go from macro to geometric description. And so the assumptions that we impose on our model are essentially, we have non-negative densities. Sure, they're compactly supported and integrable and bounded. And here we also assume that they're BV. And of course, so this is not unusual to assume, because what you have to keep in mind is that if we want to have a Mesa limit of this type, then we have to allow for these jumps here. So this is a minimal requirement. So this is not too crazy. Then for the growth rate, or the death rate, we assume that it's a C1 function that is strictly decreasing. Okay, so I've just given a cartoon here. And we assume that there is such a number, Pm, called the homeostatic pressure, where our growth rate drops to 0. And the motivation for that is that when the tissue is in homeostasis, so when it's in equilibrium, if you will, then as if the pressure is slightly larger than that, our tissue will start dying, whereas if the pressure is lower than the homeostatic pressure, then essentially there's still room for our tissue to grow, and therefore the growth rate is still positive. So this is the motivation for these assumptions. And then, concerning the drifts, and this is the novelty, we need some third derivatives in something slightly better than L2. And so this is the mixed derivatives, so we also need some minor integrability. So it's a bit tedious, but essentially some assumptions on the drift are so that we can still propagate this BV, these BV bounds, and then there's some assumptions that we really need in order to get the strong compactness of the pressure gradient itself. Okay, and so based on that, now I can present the results that we obtain. So on the one hand, we have the strong compactness of the pressure and the density itself. But that's, I guess, maybe not so surprising. What's more surprising is that we also control the derivative. So we control the gradient, and we have strong compactness in L2 of the gradient of the pressure. And so with that, then all the terms in our equation, so that we can pass to the limit in all of these terms, we can justify it rigorously, and what is more, we have these two complementarity relations, so the first one that I showed you, and then this one that essentially tells us the pressure distribution inside of our tissue. And moreover, we can show that N and P at the limit are uniquely determined quantities. Okay, so basically, I wanted to split this, just like we split our paper, into two sections, let's say. So there's one on a priori estimates that are more or less for free, I'd say. And then there's the ones where we have to work hard and where we actually found some new regularity theory. And so the ones that are for free are these L1 estimates, because we can just integrate the equation for the density. We have some L infinity control, but this is due to the fact that essentially, if we take a Barenblatt profile, so a self-similar profile, we can always put it on top of our density, and then we can control, by comparing with this larger profile, we can control the L infinity norm as well. And then we get control of the pressure gradient, which tells you it converges weakly, but not strongly. Let's keep that in mind. And this is, follows from studying the equation for the pressure itself. And then the final set of estimates is these BV norms. As I foreshadowed, that's why we also imposed this initially. But all of this is not enough to justify this elliptic equation inside of our tissue patch. And so that's why we need a second set of a priori estimates that give us the strong compactness of the pressure gradient. And so there's two ingredients that allow us to obtain that. And so the first one is an L4 estimate that Noemi and Benoit have shown or proven in a previous paper in the case of nutrients. And then we combine that with an LP version, so not an L infinity, this lower control of the Laplacian, right? But an integral version of the Aronson-Benellan estimate. That's been explored by my collaborators and I in some recent papers. And so essentially what we get, instead of having a control from below on the Laplacian of the pressure, now we control the negative part, but in some LP, so in L3 in our case, so in Lebesgue space. So essentially, the key message is we do not need this lower bound. We can also get away with some integrability of the negative part of the Laplacian. And so now we have all of these different estimates, and we want to piece them together to complete our puzzle. So from here, this will allow us to get the Laplacian in L1 uniformly. We already have the strong compactness of P, and we know that the pressure gradient is in L4. And if we combine all of these things in a good way, then this will yield the compactness of the gradient of the pressure in L2. This is what we were after, and this ultimately allows us to justify the complementarity relation, right? Only if we have this, we can pass to the limit in here. So how do we do that? Well, I've just removed all the unnecessary terms, let's say, just to give you an idea of what's going on. So I've removed the convective term, I've removed the growth term, so we've just taken the equation for the pressure, right, without growth, without drifts. And what we can do is we multiply this equation by minus Laplacian p, okay, as we see here. And then, so if we integrate by parts in here, we have gradient p dt gradient p. And so this is essentially the evolution, or d by dt of the L2 norm of the gradient. So this comes from here. And then here, we keep this term, because it's already something negative, so we put it on the other side. So this will also be controlled by the estimate. And then there's only this term left. Okay? And so if we massage it a little bit, then the final estimate that we obtain, and it's not exactly the same, because we still have to keep track of the drift terms, the growth terms. This is just purely for the diffusive or parabolic part of the equation. But you see, if you study the evolution of this first order functional, so essentially 2 derivatives on p, you get a control on essentially 4 derivatives on p, as here and in here. And so this is the first observation that we use. The first ingredient is study the evolution, or study the dissipation of the gradient of p in L2. And this gives you some control of the Laplacian and the second derivative on the support of p. And then the other one is a neat trick that Benoit and Noemi discovered. So if you take the L4 of the pressure gradient, you can write it as a gradient squared times a gradient squared, of course. And then we integrate by parts, so we move the gradient onto this guy. And if we expand all the terms, okay, so we take this, we integrate by parts, we expand all the terms, then this is what's left. And now we just do a Young's inequality, and we see that, okay, we start with the gradient in L4, we get another gradient in L4 from here, right? And this is the other term from Young's. And we get another gradient in L4 from here, and this guy. And so we subtract these two things, and what we obtain ultimately is, well, a quarter, but essentially gradient in L4 is bounded by, well, these second order functionals. So the Laplacian squared and the mixed, or all derivatives of p squared. So this is the second thing. And now we want to join them, we want to merge them. Because if you have a closer look, this term is controlled by this one, and this term is controlled by that one, because we have that the pressure is in L infinity. So it's a bit of a tricky computation, but essentially what we've obtained is we get an L4 control by studying the dissipation of the L2 norm of the gradient, which is one of the crucial ingredients, if you remember, in order to then justify that the gradient converges strongly. The other thing that we need is this Aronson-Benilan type estimate. And so historically, the classical estimate uses a comparison argument, which in our case, well, we don't want that because we have these drifts. I mean, we know what the comparison looks like. So instead, we take the equation, we differentiate it twice, so we pass to the Laplacian in the equation. And then we call W minus the Laplacian of the pressure. And then we obtain an equation for the negative part of the Laplacian. So essentially what we do is we study the evolution of a second order functional now. And what happens is that if we do all the analysis, which is lengthy, and especially with the drifts and with growth dynamics, what we observe is that this functional, so this integral, is being dissipated in the following way. Right? So we get an L3 control on the second derivatives, well, the negative part of it. And so then we can integrate it, and we see that the Laplacian of p, the negative part, is integrable in LP. And so these two things are exactly what we needed. So we have a second order control of the pressure, we have the gradient of the pressure in L4, and we have that the pressure converges strongly, and we amalgamate all of this information, and then we have the strong compactness of the pressure gradient, which allows us to pass to the limit in here. This comes from the strong compactness of the pressure, and so this is rigorously justifies this complementarity relation that governs the flow of our boundary. And so what is more, we can show also that the limiting density stays between 0 and 1, and we can pass to the limit in the equation for the density, and we have the second complementarity relation. And the final thing that we need to show is that the limit is unique, right? This is the only thing that we claimed and haven't shown yet. And so to this end, so we use a strategy that was already introduced in the paper by Perthame, Quir, and azquez, and we adapted it to incorporate drifts. So what we do is we study the difference, I mean, it's the standard thing to do, between two solutions, so n1 and n2, and p1 and p2. We test it against some test function psi, and then we just take the integrand and we rewrite it. So we factor out a z. At this stage, it doesn't matter what it is, but I just want you to focus on like copying all of these terms. We divide by z and we multiply by z. And then the last term, so we do this for the time part, the second order derivative on the pressure, which now sits on the test function. This is the drift part. And then for the growth, essentially we've written this in a very complicated way, but this guy, the gray panel and this gray panel add up to 0. The difference of the p's cancels, and so we get back exactly to this line divided by z and multiplied by z. So why on earth would you do this? Well, now we can substitute these, so we give those special names, these quantities, and then we can rewrite our equation in the following way. We factor out a z, and then we have essentially an equation for a backward equation for psi, right, with these coefficients a, b, and c. This is what we get. And now the final step in this duality trick that we use here is, well, the equation for the difference is essentially equivalent to this one. And so we haven't chosen psi, our test function, yet. We just said, let's take an arbitrary test function. Now at this stage, we take a special one, namely a psi, such that the whole equation is equal to a times yet another test function. So there's two test functions, but now we set psi to be the solution of that. So the solution of this red part, which is equal to a times psi. And all of a sudden, this complicated looking equation boils down to, well, we replace all of this by a times psi. And so since a is n1 minus n2 divided by z, we have this cancellation here. And so what we're left with is n1 minus n2 times psi. And so now this looks really neat, because this essentially gives us the uniqueness of n1, because we haven't set psi. We just said we chose any psi such that our psi satisfies this equation. Now we choose any psi that we want, and we have the uniqueness of n1 and n2. And so then the final step consists of having a look at the weak form again, using the fact that n1 and n2 are the same. So these two terms, they drop from our equation, and the only term that remains is this one, and the growth terms, like here. And so then you can see if we substitute the difference of p1 and then p2 for the psi, and we integrate by parts, we get the gradient of the difference squared. And this is equal to n1 times the difference of the g's times the difference of the p's. And now, since our tissue, our growth rate, depends on the pressure in a monotonous way, right? So the growth rate should drop as the pressure increases. We see that this term always has a sign. It's non-positive. And so we've shown that essentially a non-negative quantity is less or equal than 0. And this can only be if the gradients match. And then from there, we infer that P1 has to be equal to P2. And so there we are. This shows that initially that the equation for n has a unique solution, and therefore the equation for P has a unique solution. And so this is all that we did in this paper and that I wanted to present to you today. And I'd like to close on some future work that I want to conduct. And so the first one is a very tricky question. So during my time in Paris, actually, I've been quite involved in this Brinkmann type equation that incorporates viscoelastic effects. So now our model only has some say, friction or interactions with the extracellular matrix. Now we want to have friction between cells. And this was a model that was proposed by Bouchelet and Perthame. And so we have this, this viscosity sitting here. And now you could say, well, if nu goes to 0, right, then essentially this potential W will be equal to P. And so you would expect that from the Brinkmann law, you can go back to the Darcy law, right, but just by replacing W by P. But so this estimate requires, well, this building this bridge between these two models requires a different set of stable estimates that is, well, up to this point seems very tough. So this is something that still has to be studied in greater detail. And then also associated to this problem is the two-species version with different drifts. And so I think this is a model that I'm proposing since many years. And one thing hasn't changed. And the thing that hasn't changed is that not even existence is clear. Right? So many people have looked at this from different viewpoints. So again, optimal transportation, viscosity, certain types of approximations. But the problem is that this is really a wolf in sheep's clothing, because you have this this term essentially behaves like a hyperbolic equation, but it is parabolic. And so you don't know what tools you have to combine in order to justify, to give a meaning to the equation and to show existence. And the same is actually true also for different mobilities. If we were to include, say, if we were to assume that the cancellous tissue moves or diffuses or responds to pressure faster than the normal tissue, then I know of one existence result. And it's one dimension and for a very special solution. And the final thing is, so if we go back to the two species that I've shown here, so we have these different approaches, microscopic, macroscopic, and geometric. And so this is, again, this blob of tissue. So we really see that it behaves as a bulk. But when you have these, when the two species interact, we have these internal layers that gives you an additional, problem in terms of regularity, because now you have internal layers on top of the free boundary. And so what I would like to do is build a direct connection between interacting particles, so the micro scale, that allows you to go directly to the geometric description without passing through the macro scale, essentially. And so these are the open questions. So there's some work in this direction, but it's very, very preliminary. And OK, I guess that's all I wanted to talk about today. Of course, I would like to thank my collaborators, because, I mean, this is one work out of many with my Parisian friends here. And so, yeah, thanks a lot for the invitation. And yeah, I'd be happy to answer questions.
Just one thing, when we're looking at this last interacting species issue, the boundary between the two species, wouldn't you expect it to have some fingering showing up? How would you get it in your model? Because it's looking very regular in... Well,
so this is actually like, so we had a chat with Daniel Cohn about this, and it depends on the, so in the biological experiments, actually, so depending on the initial configuration of your cells, you have some irregularity in the boundaries. So it's actually quite interesting, because this is something that we don't observe in the model. So if your initial condition is like this, essentially initial condition for the biological experiments, then you have these cell protrusions, like these fingers that you see there, right? But then for these circular initial data, you see that the boundary is actually quite smooth.
You have fingering while you don't have the second species in front of you, but when you start interacting with it, then you sort of regularize your boundary.
Exactly, yeah. And so I don't know, we don't know why it happens that you have this. I mean, so this looks very regular in terms of like two interacting species. But you're absolutely right. If you have initial data like this, then the zigzagging behavior that you have here is more pronounced. And unfortunately, I can't explain where it comes from. But in the mathematical model, so if you just take the symmetric, so this is essentially our pressure, right? And so we've taken these initial data, and then you see that at least in terms of the model, the interface is completely straight. And so I don't know how to account for this, but maybe it's because when you pass from a stochastic macro model then you do some sort of averaging that, sometimes it's like this, but maybe sometimes it's like this. So that's why you expect on the macro scale, you don't get this type of resolution.
Okay. Okay, any other question in the audience? No? Do you have some internet questions?
The question is, are there concepts of energy in these problems which could help?
Well, so for the parabolic part, you have some energies, and also for if you incorporate drifts, right? And so, so if you have energies of this type, so some k norm. So this would be an energy that is associated to and so yeah, you're right that essentially this gives rise to a sequence of energies with a sequence of minimizers, and so then this gives rise to a sequence of problems, and this is exactly what the optimal transport community does, right? So what you do here is you have a sequence of energy functionals and minimizers, and so the natural thing to study is gamma limits, right? And so the limiting energy functional would look like essentially still phi n, and then plus an indicator, like what's it called? So essentially plus infinity if n infinity is larger than 1, and 0 otherwise. So, and this also gives you an incompressibility, or like an incompressible fluid. So, for instance, Sant'Ambrogio has been very active, and Maury in the context of traffic flow. And essentially, this pressure term then becomes the stiff pressure in form of an indicator. So you have a convergence of energy functionals and convergence of associated flows. But actually, an interesting question also is how you incorporate growth dynamics, because then you have to construct different metrics.
Okay, thank you very much, Markus. So let's thank Markus again if there are no other comments.